All right, so today you guys do have class and classwork to do, even though I am not here. What you're going to essentially do is make sure you grab a notes packet, and you still want to have your journal out because you're going to do your do not and your learning objective, and I'll just check it when I'm back. The sub or the front of the room should have the packet for today's stuff. It's going to be on transcription and translation for the central dogma. We're going to intro off again with the learning objective. It says students will be able to explain the purpose and process of transcription using models and recognize how it is a regulated process. And then you're going to answer these 3 questions from last week's content that we did. The first question is, what does DNA do? The second question, why is DNA important? And the third question, where is DNA located inside of the cell? Go ahead and write the answers to these 3 questions after you've written your learning objective by pausing this video, and then in your journal, I will check them again once I come back. So what we're going to talk about today is about protein synthesis. And you start off with DNA, but DNA is stuck in the nucleus, so it can't leave, and you still have to make things out of it because about 70% of our dry weight of our body is made out of protein. And you have to get the info out of the DNA, and it has to become a physical shape. So that's protein synthesis, or making proteins. The central dogma of biology follows this order. You start from your DNA, you do a process called transcription, and you make a mRNA, or a messenger, that is sent, and it gets translated into proteins as the end result. So you're going from DNA to mRNA to proteins, and that's overall known as the central dogma of biology. So let's first off compare DNA versus RNA, just to refresh our memories. So DNA and RNA actually stand for longer words. DNA itself is a really long word, and RNA is quite a long word as well. DNA stands for deoxyribonucleic acid, and then RNA stands for ribonucleic acid. So this is just basically saying that there's a part of the DNA that has been deoxyed, or an oxygen has been removed in comparison to the part on RNA, but the names are shortened to RNA for R, N, and A, just to make it a little bit easier for us to write and spell and say. Now, DNA is different from RNA also because it's double-stranded, and it's in a double helix. So the number of strands is 2 instead of just 1, like you find in RNA. Therefore, DNA is more complicated, and we usually keep it as our, solid structure for holding our genetic information, whereas RNA is kind of something where if you make it and you use it and you break and destroy it, that's totally fine. There are 4 bases for both DNA and RNA, and the DNA bases are A, T, G, C, adenine, thymine, guanine, and cytosine, whereas in RNA, there's actually no T, there's no thymine. We have A, U, G, C, or adenine, uracil, guanine, and cytosine. And DNA's location is generally just in the nucleus. It can't leave. The whole point is in the nucleus is that it protects the DNA, and the DNA doesn't get damaged from any outside existing forces because it has this extra layer of membrane. The RNA can be found in multiple places. It can be found in the nucleus when it's being made, and it's found in the cytoplasm as it's delivering its message that it holds from the chunk of DNA that it was copied from. So these are the main comparisons and differences between DNA and RNA that we have. Now, I do want to make a quick check, so go ahead and make sure that you do know the answers to all these questions. First one, where is DNA located? Second one, where are proteins made? Third one, why can't DNA go directly to the ribosomes and just give instructions? And then fourth one, what has to happen in order for the information in DNA to be taken to the ribosomes? I would like it if you could pause and answer these questions on your paper. You should have a section to fill them out. All right, if you tried to answer all the questions, or at least try some of them, let's go over the answers really quickly. So where is DNA located? That one's easy. It's in the nucleus, in the center of the cell. And then the second question asks, where are proteins made? Proteins are only made in the cytoplasm on the ribosomes. They can't be made elsewhere. The ribosomes are little protein factories. Now, the third one says, why can't DNA go directly to the ribosomes and give instructions? Well, we said earlier, the DNA does not ever really leave the nucleus because it's going to get damaged, or it's going to make it easily get mutations. So the DNA doesn't leave and go elsewhere. Therefore, it can't ever reach the ribosomes and it can't ever go to the cytoplasm, right? The fourth question here says, what has to happen in order for the information in DNA to be taken to the ribosomes? Well, you have to send a message then from the DNA. Note also, we're never going to send the whole DNA message out because you never need to read the entire library of information stored in your DNA. You only ever need like one library book at a time. So you'll send one little chunk of a message, like one library book, out into the cytoplasm to be read and to be have the protein produced. And then afterwards, it gets removed, it gets broken down, and that message is no more. So a message has to be sent from the DNA to the cytoplasm to get the process of protein synthesis started. So who has the instructions for making proteins? It's DNA. And DNA has to tell the messenger, which is called messenger RNA or mRNA, since DNA can't actually leave. There are two steps, like I said, in the central dogma of biology. The first step changes DNA or reads the DNA and creates a short message segment called mRNA. And that process of doing that is called transcription. Once you've done transcription, that message that's created, or the mRNA, is going to move from the nucleus, where it was created, into the cytoplasm where the ribosomes are so it can deliver its message. And once it gets to the cytoplasm, then it goes ahead and does translation with the proteins on the ribosomes, and that's how the proteins are actually made. So as an overview for our central dogma, we have transcription, which turns a section of DNA into an RNA for us to move out. And then it gets translated into a new language of amino acids where then the protein is made. Where is this happening? Well, it's going to be happening from the nucleus and leaving the nucleus and then going to the cytoplasm. Right, so this is transcription in the nucleus. The RNA is being created by RNA polymerase as DNA is open. And then translation happens in the cytoplasm on the ribosomes, which is this big blue thing in the background. And amino acids are getting built together based on the mRNA message that came out of the nucleus from the DNA. And the end product is proteins, which generally make up our physical bodies, or any organism's physical bodies. So let's talk about transcription, which is step one. We're going to talk in more detail. And again, mRNA stands for messenger RNA because it's the message that is sent from the DNA to the cytoplasm in the case that DNA can't leave on its own. In transcription, there's an enzyme that is called helicase. And it's called helicase because it's kind of like a little helicopter, and the helicopter is a chopper, and it chops the DNA by unzipping it down the middle, like this image down here. It breaks the hydrogen bonds and separates the two strands of the double helix. And so one side becomes the template, which we're going to read off of, and the other one is the complementary strand, which matches. Now, to be clear, complementary here means matching, and it doesn't actually mean like somebody saying something nice to you. Complementary in this case would be like, hey, your shoes match your pants. They're complementary. Okay, so again, helicase is unzipping, and the template strand is going to be the one that we're going to copy or make an mRNA strand message off of. So transcription will begin at a place on the DNA on the template strand called a promoter. And a promoter is kind of like the person that, if you ever go to the mall, they're kind of standing in the middle and they have a bunch of flyers and they want to hand them out to you. And they're trying to promote that you go there and get started and do something. This is the same idea on the DNA. There is a part of the DNA that has a kind of signaling thing that's like, hey, come here, do this, go over here. And the promoter signals to an enzyme to start building an mRNA after attaching at that location. So the enzyme called RNA polymerase is the one that actually builds an mRNA. Notice that it said RNA polymerase because it's building RNA. They are specifically named like that. So it's kind of, straightforward in science. So it's going to attach nucleotide bases of mRNA to the template strand of the DNA, matching it, except for the fact that mRNA rules are a little bit different from DNA rules, right? In DNA, it was ATGC. So A and T paired together, C and G paired together. But in RNA, A actually pairs with U. So if you ever see a T, you automatically know it's DNA. And if you see U's for uracil, you're actually going to say that it's mRNA or just any RNA. So in this case, A will pair with U. If you have a T in the DNA, it will pair with an A. If you have a C, a G, and then it will pair with a C. The bottom rules are pretty much the same. The only biggest difference is that the A on the DNA will end up pairing with a U on the mRNA. Now, this might seem kind of strange, but it always has worked, so it's fine how it is, right? And I want to be clear that not all genes are expressed. We've talked about this before, like having a whole library of books and only taking 1 or 2 books at a time. This is true. In fact, even inside of the books, not all the books are read, right? Sometimes when you read a book, you skim through some areas and you read the parts that you actually really need. That's the same idea too when we're talking about genes being made into messages and then only the important parts being kept. So this says eukaryotic genes contain regions that are not translated into proteins. These regions of DNA are called introns, and they must be removed from mRNA by a process called RNA splicing. So the useless stuff gets chopped and then it gets removed. So the message is as concise as it can be. For example, if you have like Twitter or when Snapchat, I guess, had like a limit, you can only write out a certain amount or you can only send a certain amount of characters. So the message has to be as concise and as direct as possible. And this is kind of a similar case. The remaining portions of DNA that are translated into proteins are called exons. Think exons, expressed, the important part that is being shown or expressed as the physical trait in the protein. So another quick check, go ahead and make sure that you can answer all these questions before you move on. Why does transcription happen? Where does it happen? Why must it happen there? How is mRNA made? Where does the mRNA go after it is made and why? And then what is RNA splicing? Pause and try to answer these for yourself. Okay, so why does transcription happen? Well, DNA's got to end up making some proteins, right? So the first step is you got to send a message because the DNA can't leave. Where does it happen? Well, DNA is in the nucleus and it's not leaving anytime soon. So obviously transcription happens in the nucleus. Why? That kind of answers the third question, honestly. The third question says, why must it happen there? It's because DNA is there. How is mRNA made? Well, DNA is unzipped by helicase. RNA polymerase goes in and on the template strand, it's going to start building an mRNA strand. mRNA polymerase will come in and start building the mRNA strand. And then the mRNA will double check, detach, and then leave. Where does the mRNA go after it is made and why? It's got to go to the cytoplasm because the ribosomes are in the cytoplasm. That's the only reason. Ribosomes aren't in the nucleus. You can't build anything there. And then what is RNA splicing? RNA splicing is when the information that is not wanted, aka the introns, are deleted from the mRNA. So only the important message with the exons that are about to be expressed are left as the message is sent from the nucleus to the cytoplasm. So let's practice some transcription. I did say earlier that we're matching letters now. There's no T's in mRNA. There's only U's instead of T's. So A will match with U. And then if you see a T, you're going to match it with an A. And then a C and a G are still matching each other. So if we have a DNA right here, and we have ACC, AAC, TAGC, we should be able to then go ahead and match each letter with its complementary base in the mRNA rules. So let me go ahead and insert a text box so I can write here. And so A, like we said, doesn't match with T anymore in mRNA. It matches with a U. C matches with a G. C matches with a G again. A matches with a U, like we said. A matches with a U. C matches with a G. And then we have a T here. And T can still match with an A. And you say, Miss Sun, well, we weren't supposed to have T's, right? But keep in mind that the strand above saying DNA is still allowed to have T's. But when we are copying into mRNA, like this next letter, we cannot have any T's in it. Instead, it should be a U that matches with the A. And then we have a C and a G to finish that off. So that makes our complementary mRNA strand for the above DNA strand. I'm going to ask that you follow the rules A, U, G, C, and make the rest on your paper into mRNA strands for me. The next thing you're going to do after that little bit of practice is you're going to do a little bit more practice. And what you're going to have is you're going to go to a Google Classroom. You're going to open up the interactive notebook for week 20, which is the current week that we're on. And you're going to complete the first section as your independent practice. Now, you're not going to turn it in yet because I still have you technically another day of this week. And you're going to go ahead and just leave it as once you finish the section. And at the end of the week, you're going to click turn in on Google Classroom and I'll grade both sections for you. At the end, once you finish the independent practice, you have a quick exit ticket. It's going to be a Google form as well on your Chromebook in Google Classroom. And you're going to finish that, and then you'll be done to chill and relax for the rest of the class period. That's all I have for today. Good luck with all of your work, and I hope to get it from you so I can grade it for you.